So to start off, I'm going to do something a little bit different. We're going to do something called a think, pair, share. So I'm going to present to you guys a question. I want you guys to tell me what is the big picture of intracellular membrane trafficking. I think everyone's back in here. Yeah. So now that you guys have had like a minute or two to discuss the big picture of chapter 13, let's hear like one person from each room just give us what you guys came up with. There's no wrong answers. This is super open-ended.
What my group said was basically the chapter was about how substances like proteins and macromolecules travel from the endosome, or the ER, the Golgi, the endosome, to the plasma membrane through vesicles, and then the aspects of what control the directionality of it.
That's awesome. Let's hear from group two or one of the other groups.
So I was just gonna say my group was struggling a bit with the question. We were basically just talking about like we know that there are mechanisms of like membrane transport and that's involved in cell signaling, but that was kind of all we got.
That's great. Yeah.
Awesome.
Yeah. And the coating will disassemble, and that leaves you with our finished result, which is a naked transport vesicle. And what's one place a vesicle can go after it's naked?
Early endosome.
Great. Anyone else? What else? Where else can it go besides an endosome, early endosome? Where do vesicles go? Sage?
The vesicles?
Yeah, where can vesicles go?
They can go to like the Golgi, right?
They go through the Golgi. Okay. What about K Wisdom?
Honestly, I'm not sure. I'm looking through the PowerPoint right now to kind of like figure out, but I'm a little bit lost.
No, you're good. You're good. That's what we're here for right now, right? To make sure this makes sense. So we're going to talk more about where vesicles go. I just kind of wanted to popcorn some ideas out there to see where you guys are at, just kind of get a sense of what you know. Okay. So we're good there. Let's go on to, that was our steps, right? And don't worry, I'm going to send you guys this all typed up by Friday. So,
awesome. Thanks.
Yeah. So I heard someone mention this protein for number 4. What protein makes the final pinch of a budding vesicle?
Dynamin.
Dynamin. Perfect. Everyone's saying dynamin. Okay. So how does dynamin work? It's a protein, right?
Right.
So what are its main energy units that it uses to turn on and off?
The GTP and GDP?
Yeah. So GTP and GDP, you're going to hear, as long as you guys stay within the realm of molecular and cell biology, like that's bread and butter, right? So it's similar to ADP and ATP. Those are like your two bread and butters, your meat and potatoes. So usually, like 99% of the time, GDP is off and GTP is on. And there's a monomeric enzyme called GTPase that is doing the constricting of the membrane neck. And if you guys want to see how this works on YouTube, there's a great animation that like these new high-tech microscopes got of this enzyme actually making the pinch and releasing a budded vesicle. This technology is getting really cool. So any questions about dynamin? Clear on that? It's a lot of proteins coming at you guys. I just want to make sure we don't move forward without making sure we're clear on each protein. Okay. So number 5, what proteins assemble COP1?
The ARF protein.
Does everyone agree with her? ARF. So we say ARF. We have a lot of weird names like that in biology. Not sure how that one got its name. So ARF proteins assemble COP1 in Clathrin coats at the Golgi. What about COP2?
The SAR proteins.
Everyone agree on that? So SAR proteins they control assembly at the ER membrane, right? So we have these two routes that these vesicles can take, we know, Golgi and ER, COP1, COP2. I bet there's going to be questions on the test that try to reverse those two. So don't get SAR and ARF confused with COP1 and 2, right? Okay, so keep that in mind. All right, so now we're going to go over another mechanism. GTP hydrolysis by SAR1 leads to what?
I put D.
We have 1D. Let's hear from everyone.
I agree with D.
D? 2 Ds. So D is the right answer. And you guys are my only PLF section who got that right this week. So great job. You guys are ready for the exam? So yeah, GTP hydrolysis by SAR1 led to disassembly of the COP2 protein coat. How'd you guys get that answer?
So I know that when SAR1 has GDP bound, the diphosphate, it's inactive. So when it has GTP, the triphosphate is active, being able to bind to the membrane and assemble. But when you hydrolyze GTP to the diphosphate, it no longer is going to be active. It's going to disassemble itself. I mean, that's what I thought.
No, that's a perfect explanation. So why does it need to disassemble?
It's not going to be able to bind to the membrane anymore, right?
Right, because it has to be naked, right? Yeah, so your end goal is to be naked. So you need something like D to occur of that protein coat. And the way it works is the GTP hydrolysis by SAR1.